The overall goal of this procedure is to produce active Adeno-Associated virus serotype 3 vectors which can efficiently transduce human liver cancer cells. This is accomplished by first transfecting 3 different plasmids into 293 cells simultaneously. The second step of the procedure is to purify AAV3 virions assembled in 293 cells. The third step of the procedure is to determine the titer of the purified AAV3 vector stock. The final step of the procedure is to transduce human liver cancer cells using purified AAV3 vectors. Ultimately, results can be obtained that show highly expressed EGFP protein in human liver cancer cells through fluorescence microscopy.
The main advantage of this technique over existing methods is to achieve high efficiency of gene transfer and expression in human liver cancer cells.
Though this method can provide insight into the production of AAV3 vectors, it can also be applied to other systems, such as more widely used AAV2 vectors.
Generally, individuals new to this method will struggle because of the involvement of a number of steps which can be tedious.
Demonstrating the procedure will be Wenqin, a biological scientist from our laboratory.
The experimental manipulations in this section of the protocol are performed inside a tissue culture hood. 48 hours before beginning this procedure, seed 10 15 cm tissue culture dishes with human embryonic kidney cell line 293 so that the cells are 70 to 80% confluent on the day of transfection.